Roshan Tiwari has obtained his PhD in physics recently from the Indian Institute of Science, Education and Research, that is ISER, Kolkata. He completed his BSc in physics from BHU, Banaras Hindu University, Varanasi in 2014. His research interests include spectroscopy, bio-inspired waveguides, microscopy, sensing, optical trapping, and statistical analysis. He is also interested in the connections of unexplored aspects of reality with age-old wisdom. So without any further ado, I would request Dr. Roshan Tiwari to come over to the stage and start his talk. Thank you.
Yeah, thank you, Manas, for this introduction. So good afternoon, everyone. And I'm very thankful for Bhaktivedanta Institute to give me this opportunity to present. It's not my work, but I have worked a lot of time on light. So I have tried to make a connection between memories and light. And as the topic suggests, does light affect memories? So it really seems very unobvious that how can light affects our memory? It's very difficult because memory seems to be somewhat very unphysical some things. So, but the work has been done in this area, and I will try to present that work. So I would like to also dedicate my work to my spiritual guide, His Holiness Bhaktivedanta Swami Maharaj, and also Dr. T.D. Singh, who is the founder of Bhaktivedanta Institute, for being my inspiration. Yeah, so mainly I would introduce you to the work, which is how light is connected to memories. Then yes, and the area which is mainly concerned with connecting this light with genetics, it's very important, and I will try to give you some account of this. And then memory recall and freezing by tone, because in previous talks we have heard about this how memory can be recalled by giving some vibration, some tones to dogs. They think that these tones are acting as food. So in the same way, I will introduce you to this memory recall. And freezing is like in rats, they freeze whenever they become fear. So that thing. And then ultimate thing, finally, by light, how light can act as a stimulus to create this memory recall and freezing behavior. And the parameters affecting this freezing, whenever these rats become fear. So what are the parameters by which we can create more freezing of them? That is, they will recall more and more memory of that. And finally, the summary. So as you know about light, that light is an electromagnetic wave, and there is an electric field, oscillating electric field and oscillating magnetic field. And because of this coupling, there is a directionality in that. Light propagate in certain direction. And it has a plethora of properties, which makes it very unique. Like we have a wavelength. It's a very crucial properties. By changing the wavelength, we can change the color, because the color is dependent on the wavelength. Similarly, intensity, phase, polarization, coherence. So all these properties in the last some decades have made optics light as a tool to explore the world which are not accessible to us. So like our nano world to macro scale, because a lot of reality is there which we cannot probe, but just by using light, because as you can see, I have pointer and I'm just putting laser here. So it's a light and it can be used for various purpose. So we can use different wavelengths of light to explore the world which is not accessible. Even atoms, we can understand about atoms, molecules, cells, and even we can go beyond our this terrestrial environment. We can go to galaxies. We can get a radio telescope to explore about what is there outside. So that's why light is very important, and that's why I'm very interested to use light to explore memory. So the question is, can we probe memories using light? So as we know that memories are not like physical things. In order to interact with some light, we need some objects, because light finally has to interact with that object. But when we think about memories, these are like photographs. What episodes in our lives store in some sorts of photographs, how they can interact with light. You can see a person having images of all his childhood's sleep like this. So it seems very unobvious. So before going further, I will try to we will try to learn about how things are stored in our brain. So we all know that we have five senses, and these all five senses are they get the information from the surrounding, whether it's a light information, whether it's a sound information, whether it's a taste information, and finally they convert it into electricity. So that is very amazing, because we are also using electricity, and our body also using electricity as an efficient device to communicate. So that's why I found electricity very puzzling and sometimes very interesting also, because and so all these sensory informations go inside the brain, and then the brain start processing. This all we know about. And the neurons are very special because they are the electrical wires. Like we have electrical wires, they are electrical wires in our entire body, which conducts electricity, which conducts entire information to the brain, which process it like a CPU. So you can see it's a neuron, and you can see the electrical burst which is going on in order to communicate. And you know, it's 86 billion neurons in our brain, in our body, and there are 200 billion stars in a universe. So you can see it's already comparable. It's just double, but still we can see the entire web of complexity in our brain. So memory storage, where memories are stored, especially I am concerned with the fear memories. So you can see it's our brain, and there is a hippocampus there. In previous lecture, Jayasida Sharma ma'am has told about hippocampus, and in hippocampus there are certain type of areas where you can see this is dentate gyrus, and this is another area where the memories are stored. So especially in dentate gyrus, people have done a lot of research, and they found that the fear memories are stored there. And so there are three parts: dentate gyrus, subiculum, and CA field, and they form these trisynaptic circuits, which are responsible for processing and storage of long-term memory. So n-grams are basically interesting because these are type of some types of, you can see these are the, if you consider these spheres as neurons, so the collection of certain type of neuron give rise to certain memories, and different type of memories give rise to excitation of different these blocks. So it's a simple model by which we can understand how memories are. So when n-gram state is like when some memories get activated, so they are all these red blobs will become correlated. But when they are in dormant state, they are not correlated. So this memory is also very dynamic. Sometimes they become correlated. Sometimes when I see my dream also, sometimes I see a horse body but elephant head. So how they get connected, I have seen many times. In the memories they are coming. Unphysical things are coming. It means that some things get connected, they are correlated. So now the question is there a way to excite this neuron specifically? Like if we want to specify only certain neurons, can we do that? And is there a way to control the behavioral output? If I excite certain neuron, that then I can control the behavior of some sorts of. So the answer is yes, optogenetics. In the last 20 decades, in the last years, a lot of research has been done in this field where they are using optics and genetics in order to probe what is happening inside and how to control the behavior. So optics, I know some sorts of optics, but genetics is somewhat new to me. And in the last few months, I have read about it. Very interesting. I found it very interesting and very mathematical also, because genetic engineering, it's very mathematical. How genes are manipulated. So let me give you introduction to genes. So genes are basic and fundamental unit of heredity. For example, when we see child and father, we found a lot of things common. Their faces are common. Even their walking style sometimes we find very common. So is there a gene responsible? So the gene is the fundamental unit. And you can see there is a nucleus inside the cell. There is a cell and there is a nucleus. And inside nucleus, there is a chromosome. And then inside the chromosome, genes are certain segments of DNA, which is responsible for certain properties, certain features of an individual. And genes are made up of DNA. Because the DNA has 4 bases, because of this permutation combination, different properties get manifested. So some genes act as instructions to make molecules called proteins. So these genes have used to do a lot of things. So one of things is that they make proteins. They give instruction. They are like a code. They give instruction to make certain proteins at certain amount of time if a body requires. So So there are 6 steps of optogenetics. First thing that you need to have, for example, this optogenetics is about using genes which are light sensitive. So these are the genes which are light sensitive genes, and these are attached with a promoter to drive expression. So these type of genes has to be expressed at a desired time. Another thing is that these genes have to be instructed into a virus. I will tell you all these steps. And then what they do that they, when the virus become infected with this type of genes, they infect the virus inside a mouse. And then what happened when they infect the virus with with this type of protein? This type of protein expressed in targeted neurons. For example, we want fluorescent neurons inside the brain. Then these genes will go there and they will sit on that. And then this type of genes will be expressed. And now we use light to control the mechanism, to control the behavior of rat. Because what happened finally, this type of genes, these are called light sensitive genes. They are very sensitive to light. So when the light is put, using the optical fiber, you can see they are putting light and then they are exciting this type of protein molecules. So these protein molecules are channels, ion channels. So when they open, certain ions will go, because finally our neurons are ion channels. Our neurons are conducting charged particles. So if we control the charged particles, we can control the neural activity. So this is very important. That the gene, this type of channel rhodopsin protein, they are expressed in the mouse. And what are they? This is a neuron, and at certain portion they get expressed. And this is that channel rhodopsin gene. This is a very nanopore. The size of this is very, very small. And certain, then if a light falls on it, then they get open and ions will move across it. And this will create an action potential and certain neural activity will happen. So without it, you cannot change the neural activity. But now using these channels, you can change the neural activity. So this is very similar, like no light, channel rhodopsin inactivated. Neurons doesn't fire, but as we put a blue light, because they are excited for particular wavelength, only blue. So they get open and then now they are the ions are moving across it and they are creating a neural firing inside the mouse, you can say. So they are wavelength sensitive and they create a spike, and the important thing is that they are acting like a switch. Now we can on off, we can do on off thing here. When they are on, certain neurons are excited. When they are off, it's closed. So we so scientists have like somehow they got some control over switching the neurons off and on. So this is the work. This is the scientist from MIT who did this work in 2012, and this is a very basic paper, but from that a lot of new avenues have evolved. So now I already told you that in previous work many conditioning type of things talks have been done, like giving a tone to dog, and dog is thinking that this tone is now a food. So this type of conditioning we can give to mouse also. Mouse can we can make it illusioned. So so it's a very similar experiment. Here the mouse are trained. You can see there is an electric shock there. So they are giving electric shock to mouse, and at the same time they are blinking some buzzer. So whenever the mouse gets shocked, this buzzer will start. So now after some time they are consciously thinking that now whenever this buzzer rang, there is no electric shock, they have been transferred to some different place, and whenever this buzzer rang, this fridge, they fear. So initially they are fearing because electric shock, but now they are trained to, even without electric shock they are fearing. So they now are conditioned to become fear. So certain amount of maybe chemical change this is inhibiting inside it, this buzzer. So this suggests that these neurons were involved in creating fearful memories. Because these shocks have excited some neurons, and at the same time this buzzer, and now without this electric shock, this type of neurons are getting excited, so creating a fearful memories. So the reason the researchers told that this dental gyrus, as I already told you, that these contains the fearful memories, and this is the different type of neurons in the dental gyrus are excited now. So same environment activates the same population of DG cells, because if the environment is same, certain genes will be excited, certain cells. But if we change the environment, the cell number, the cell nature will same. The different type of cell will be excited here. So it's a fear gene expression. So I would like to tell you that this is our dentate gyrus, and you can see this green one. So it is without any electric shock, without any conditioning. The brain, because inside a neuron, it's a very dark place. So in order to see something, we have to add some fluorescent material which will emit light after. But now after, after the rat become feared, you can see the intensity of green color has changed. It has become more greenish. So somehow they are able to go inside the dentate gyrus, inside the brain, and they are putting this optical fiber to get this information, or something is excited. When they get feared, this type of things evolve, this type of thing fluoresce. So this channel rhodopsin EYFP expression, they are getting expressed due to fear. These are some type of genes which are getting expressed. So now the question is, like, we have already tried with buzzer and all these things, but is it possible to recall the memory? Because finally the rat is recalling the memory. Initially it got trained whenever the buzzer is ringing and this shock, but now whenever this buzzer is ringing, it has that information of fear. It is recalling the memory. So similarly, can we do that with light? So the answer is yes. They have done that. This is a rat, and they put optical fiber there and particular wavelength, and then they did the experiments where they are delivering light to the DG region. DG region, that the same thing, that dentate gyrus thing, and they reactivates the fear memory. That fear thing, they reactivated it, and the animals freeze again, even in the absence of tone, where there is no sound. Just by light, they are making it to fear. So this is a general steps of labeling. As I already told you that there is a training phase when there electric shocks is there, and whenever there is electric shock, certain neurons will be activated. It's not like all neurons will be active, certain neurons which are responsible. And what they're doing, they are using this light now to excite that particular neurons to make it freeze. This is a video. Shravanji, you can play this video. So in this video, you can see this is a rat and this is optical fiber. There is no light now. So they are doing their normal activity, roaming here and there. So now the light is on, and still they are roaming, but let's see. So somehow they are getting feared. Somehow this light has an effect on their brain and neurons part. So they got feared and they are freezing. It's not like that they are total freezing. Some movement will be happen because some error will be always there. So as you can see, I have already told you about memory recall. They are recalling the memory, but is it possible to change the degree of memory recall? Because sometimes we don't able to recall the memory of past event.